Welcome, everybody, and thank you for joining us for today's seminar. Before we get started, I have two housekeeping announcements. The first one is that if you have a question for today's speaker, please enter it in the Q&A box, not in the chat function. We'll have time to address them at the end of the presentation. And the second one is that we will not have a seminar next Monday, November 24th, due to the upcoming Thanksgiving holiday, but we'll resume our seminar series on Monday, December 1st. So now it is my great pleasure to introduce our today's speaker, Dr. Christina Camell. Dr. Camell is an associate professor at the Masonic Institute on the Biology of Aging and Metabolism and a faculty member at the Department of Biochemistry, Molecular Biology, and Biophysics at the University of Minnesota. Her lab focuses on fundamental questions addressing the biology of aging, particularly on the role of inflammation in promoting age-related diseases. Dr. Camell is particularly interested in the visceral white adipose tissue, an organ that shows early signs of immune activation during aging. Her research has revealed distinct roles for immune cell subsets in the adipose tissue that become immunosenescent-like. A main emphasis of her lab is an optimized mouse model of the humanized immune system, which exhibits the diverse and mature immune system seen in humans. Her work has been published in Science, Nature Aging, Cell Reports, and Immunity and Aging, featuring a lot of these results. And lastly, Dr. Camell is the recipient of several awards, including the 2025 Glenn Foundation Discovery Award from the American Federation for Aging Research for her work on macrophage inflammation, cellular identity, and health span during aging. So please join me in welcoming Dr. Camell today for her presentation entitled Immune Aging and Adipose Tissue Homeostasis During Aging. Thank you, and Dr. Camell, the floor is all yours.
Thank you. Yeah, thank you so much for the introduction and for the invitation. It's really nice to be here to present an unpublished project, but it's just about to come out in Nature Aging and some more past projects from my lab. So I wanted to start off just by giving you an outline where I've broken this talk into two parts, where the first part is going to discuss the importance of inflammation and immune dysfunction during aging. In particular, I plan to focus on macrophages that are well known to become hyper-inflammatory during aging, although the mechanisms and how they maintain this inflammatory process is not really well understood. And so I'll discuss our findings in understanding how macrophages maintain their inflammatory state. And in part two, I want to discuss the consequence of the aged immune system on adipose tissue metabolism. White adipose tissue is an organ that is important to maintaining metabolic function and maintaining immune function. It is also an organ that shows signs of aging prior to the aging of other organs. And we have identified important immune cell adipocyte crosstalk that is causing adipocyte dysfunction. And so in particular, I'll talk about lipolysis and the importance of adipocyte lipolysis during infection. So why do we care about aging, and why is studying aging actually critical for the field? We know that people are living longer, so this is a map that shows the proportions of people that will be old, so 60 years of age or older, across various countries in 2015 out to 2050. And you can see that there are a substantially larger proportion of people of old age by 2050. This, of course, older age is associated with increased risk for metabolic and susceptibility to infectious diseases. It is also creates more financial burden on families and more stress among families. So the negative consequences of age and age-related diseases are multifold. And the aging field has separated our understanding of aging into the pillars of aging, where dysfunction is the underlying theme. And we appreciate that the dysfunction seen in these various pillars all contribute to a biology of aging, where the end goal is that we can understand the process of becoming old so that we can treat age as its own state and understand it as a different state from that of the young individual. And we are in particular interested in the immune system and the development of chronic inflammation to the biology of aging. And I wanted to give a brief reminder of what the immune system is and why it's important. So it's important because it provides defense against exogenous, so viruses and bacteria, and endogenous stimuli, which can arise during cellular stress. There's an innate arm, so which provides the initial and non-specific response composed of macrophages, which I'll talk a lot about today. They are very important in producing cytokines and growth factors. They're also important for their ability to phagocytose products, produce antimicrobial products, and activate complement. Dendritic cells or macrophages can be important in antigen presentation, which lead to the activation of the adaptive immune system, which provides the secondary and a more specific response. And T cells and B cells are important here, where they can develop memory and produce antibodies and actually clear out exogenous stimuli. I want to acknowledge that the variables in my projects are quite complicated because we do think about the activation of the immune system during cellular stress, so during aging, and what sorts of accumulating damage associated molecular pattern can drive the activation of this innate response. But I'll talk a little bit about infection, which develops during the context of exogenous stimuli, and whether the immune system is actually functional in response to a new stimuli. And what we know is that the aged immune system is absolutely dysfunctional, and this leads to more susceptibility of older individuals. So this is a schematic to help describe the contribution of inflammaging to reduced health span and increased morbidity of aging, where chronic stimuli that arises from cellular stress supports the development of inflammation and the activation of the immune cell, leading to more cellular pathology. And this feed-forward circle creates a term called immunosenescence that is really referred to as the dysfunctional immune system that has a dysfunctional response to new pathogen. All of this supporting reduced health span and increased morbidity. I want to acknowledge that the immune system is quite complex, where this is a schematic that describes our known findings, the field known findings about the changes in the immune system with age. And so there can be changes in B cells, in T cells, in macrophages, and all of these are really dependent upon the subset of cell that you're looking at. So I introduced the idea of immunosenescence that indicates impaired function within immune cells from older individuals. This is distinct from the development of cellular senescence, where cellular senescence is the accumulation of senescent cells that have stopped, that have undergone cell cycle arrest and have changes in the genomic DNA, but also changes in secreted products and proteins that they secrete. Major signs of immunosenescence and immune aging include increases in memory lymphocytes, so an increase in memory T cells and B cells, an increase in low-grade chronic inflammation, so an increase in cytokines that are circulating throughout the body. These immune cells do develop various phenotypes that support either their hyperactivation or their reduced activation. And we use words like exhausted, senescent-like, or inflammatory to describe those phenotypes. Immune cells also show altered polarization or differentiation. And so just to sort of reiterate some of what we see in the immune system, where in the young immune system, we describe it as naive and capable, but with aging, it becomes more inflammatory and exhausted. I'll focus on macrophages here today, where we know that they're important in multiple tissues, so not just in lymphoid organs, but also in non-lymphoid organs like the adipose tissue, where they perform tissue surveilling jobs and they're anti-inflammatory. T cells, I have one published project on CD8 T cells that are important in clearing out virus-infected cells, and I'll just briefly mention this project. But with aging, macrophages become more inflammatory. The NLRP3 inflammasome pathway has been well described to drive that inflammation, where the NLRP3 inflammasome is an intracellular receptor that recognizes the accumulation of DAMPs and PAMP and promotes the secretion of IL-1 beta. CD8 T cells become more exhausted because they express a receptor that blocks T cell receptor activation. And when they're exhausted, they have impaired functional responses, including reduced granzyme B and target killing. So one thing that's not really well understood is whether immune function in old organism can be improved. And when I started my lab in 2019, I was involved in a couple of projects. My lab led a couple of projects that tried to address this question. So these are two published projects where we used young and old mice. And when we exposed mice to pathogen, we saw 100% lethality where the young mice were able to survive. In the first project, we gave the old mice senolytics, and we saw that the senolytics were able to increase the survival of the old mice to the least lethal pathogen. And this is through the development of adaptive immune response that led to the generation of protective antibodies and survival. Subsequently, we asked whether we could actually directly increase T cell activity by giving the old mice checkpoint blockade to increase their granzyme B production and increase their ability to kill virally infected cells. In the old mice that were given checkpoint blockade, so in this case, anti-PD-1, we saw increased survival and increased granzyme B, including ability to clear the virus and actually survive. So these projects really suggested that elimination of critical cell types, so for example, senescent cells, or targeting specific age-induced pathways, for example, in the exhausted T cells, are actually realistic strategies for improving the immune dysfunction. And so this sets us up, I think, with justification in investigating different pathways that might improve immune dysfunction. We're doing this through two different ways. The first of which I'm not going to talk about because it is much too early, but this is in collaboration with Steve Jamieson, who is leading a PO1 at the University of Minnesota, where we're really investigating the consequence of the accumulating senescent cells on immunity, in particular during infection, and the consequence of accumulating secreted factors on the immune cells and their ability to clear viruses. What I am going to talk about today is a new pathway that we've identified in regulating macrophage hyperinflammation. And so this would be a project that focuses on the cytokine GDF3 that is increased in old organism and is supporting the inflammatory pathway of macrophages via what we've identified as SMAD2/3 and increased chromatin accessibility. Since macrophage production of cytokines like IL-1 beta is actually really well understood and known to be driven through the intracellular receptor NLRP3 inflammasome, a question that I asked as a postdoc was if there was any age-specific macrophage signals that accompany chronic inflammation that is known to be regulated by IL-1 beta. So this is just to show the final product prior to showing you the data that led us to the schematic. This project was led by a PhD student in my lab, so Inhua Zhang, and where she has identified GDF3, so this is a cytokine from the macrophages, as an age-specific signal that is enforcing inflammation in the macrophages via phosphorylation of SMAD2/3 that is causing increased chromatin accessibility, possibly through limiting and de-repressing the chromatin and removing PRC2, removing trimethylation of H3K27. We're excited about this because the impact of macrophages and the impact of macrophage inflammation on multi-organs is well understood. We think this points towards a new pathway, the GDF3 SMAD3, and possibly regulating chromatin as a new candidate for actually therapeutic targeting. So the data that led us to this model. As a postdoc, I isolated macrophages from young mice, old mice, or old mice that are NLR deficient in the NLRP3 inflammasome. And we identified GDF3 as a top regulated gene, so increased with age and down in the NLRP3 deficient mice, indicating that GDF3 is expressed by macrophages and upregulated by the NLRP3 and age. It's what has been described in the literature is that insulin or IL-1 beta can actually increase GDF3 expression, where GDF3 is produced as a cytokine binding activin receptor and causing the phosphorylation of SMAD2/3, where it can regulate cell fate. It's also been shown that GDF3 promotes insulin resistance in white adipocyte. However, since we know that macrophages exhibit heterogeneity, the first question we wanted to ask is what subset expresses GDF3? To answer this question, we isolated stromovascular fraction immune cells from young and old mice. So there were a number of substantial differences when we performed single-cell RNA sequencing of the immune cell, but I want to highlight the differences in the macrophages, so the 4 clusters of macrophages here. And when we quantified the different proportions of macrophages, ATM1, ATM2, ATM3, and ATM4, we found that ATM2 and 3 were increased in the old mice, where ATM1 was decreasing in proportion in the old mice. We asked what the transcriptional profile of these macrophages was, and we found that ATM2 and 3 exhibited an inflammatory phenotype expressing IL-1 beta, IL-18, they expressed ITGAT, that is the gene for CD11C, and other inflammatory markers like CD9 and MHC2. These macrophages were also abundant in GDF3, suggesting that the inflammatory macrophage is producing GDF3. And ATM1 that is decreasing with age was a more anti-inflammatory macrophage expressing MRC2 and LIVE1. So to address the role of inflammatory macrophages during aging, we use a model of naturally aged mice, where young mice, so defined as 3 to 6 months old, or old mice defined as 20 to 24 months old, are given either left unchallenged or given LPS, an activator of the TLR4 to amplify inflammation. We can analyze and quantify macrophages by performing flow cytometry, where here we can gate on the pro-inflammatory macrophages, the CD11C positive macrophages, or the anti-inflammatory macrophages, the CD206 positive macrophages. And when we quantified the CD11C positive macrophages, we found an increase in the frequency of those macrophages in the PBS injected mice, and this increase was maintained under LPS injection. So this indicates that our flow cytometry is recapitulating what the single-cell RNA sequencing is showing, and that the macrophages are becoming more inflammatory with age. We also found an expression of CD9 and MHC2 on these macrophages, on the CD11C pro-inflammatory macrophages, indicating that, which is consistent with the single-cell RNA sequencing. We have previously shown, and this is consistent with the literature, that old mice exhibit increased disease severity and a cytokine storm during endotoxemia or sepsis. And so we've done this using this model of LPS or cecal slurry to mimic sepsis. And so we can measure disease severity by looking at body temperature, where young mice maintain their body temperature under LPS condition, and old mice become hypothermic during LPS injection. We can also look at various cytokines, and we see that the old mice exhibit a cytokine storm, whereas the young mice do have increased cytokines, but this is to help clear out the LPS or sepsis infection. So the overall hypothesis for this project was that GDF3 is increased during aging, driving activation of the SMAD pathway and contributing to the development of these CD11C pro-inflammatory macrophages, ultimately supporting worsened endotoxemia during age. We have a GDF3 global knockout and a myeloid-specific knockout where we have crossed GDF3 flox mice to a lice M-Cre. We aged these mice out to almost 2 years old. We perform metabolic profiling as they age. And then at a terminal endpoint, we can inject them with LPS to amplify the inflammation and analyze the macrophages. In particular, we focus on the adipose tissue because this is an organ that shows immune aging early in life and because it is important for the metabolic function that we're interested in looking at. So this is data to show you that GDF3 is increased with age, so 10 to 20 fold, and is not present in our GDF3 knockouts. When we analyze the proportions of the inflammatory macrophage in the old wild type and knockout mice, so we found an increase in the pro-inflammatory macrophages that failed to happen in the old GDF3 knockout mice. And we also found that the GDF3 knockout mice were protected from disease severity during that LPS injection. And we found a consistent theme in our myeloid-specific knockout at 22 months of age, where GDF3 is deleted, completely absent from the adipose tissue of these mice. We also found a reduction in IL-1 beta and other cytokine, a reduction in the pro-inflammatory macrophages, and protection from that disease severity, so increased body temperature at 4 hours after LPS injection. We did find that GDF3 deletion has no effect in young mice, and this is even when young mice are given a lethal dose of LPS. To ask how GDF3 is acting on these macrophages, we analyzed phosphorylated SMAD2/3, and we found that SMAD2/3 signaling is increased in the old wild-type mice and significantly reduced in the old knockout mice. We used phosphoflow to analyze whether GDF3 was signaling through phosphorylated SMAD2/3 in the pro-inflammatory macrophages and the anti-inflammatory macrophages. We found increased phosphorylated SMAD2/3 in the pro-inflammatory, but not the anti-inflammatory. Ask whether GDF3 can directly signal through those macrophages. We isolated adipose tissue macrophages and treated them with recombinant GDF3 in vitro. Recombinant GDF3 increased phosphorylated SMAD2/3 in both the pro-inflammatory and the anti-inflammatory, but it increased it further to a bigger extent in the pro-inflammatory. Recombinant GDF3 also increased the proportions of CD11C positive macrophages, suggesting that the inflammatory macrophages are not only producing GDF3, but is signaling back on to itself in an autocrine manner. So this is a model so far where this is an inflammatory macrophage, and insulin potentially could drive the expression of GDF3. It is produced as a cytokine and signals through the activin receptor, driving SMAD2/3 and cementing the inflammatory phenotype of that macrophage. We've shown that this pathway is important in cytokine production and disease severity in LPS challenge in old mice. We've also used various reagents to block this pathway in old wild-type mice. So in particular, a therapeutic specific inhibitor of SMAD3 that blocks the phosphorylation of SMAD2/3, and the BET inhibitor, JQ1, that blocks BRD4, which is a cofactor driving GDF3 transcript. And importantly, these in vivo treatments mimicked the GDF3 deficient phenotype. We've also used human adipose tissue or the atherosclerosis risk in communities study. That is a longitudinal study following human patients over several decades. And we used the serum samples from this study to address whether GDF3 is important in age-related human inflammation in human. we found that GDF3 is correlated with inflammation in the bat and in the serum of these samples. And to show you an example of what some of this data looks like, so in the CYS3 or the inhibition of SMAD3 experiment, we take young or old wild type mice. They are given either vehicle or the inhibitor, CYS3, for 1 month prior to the LPS injection. And we see this increase in pro-inflammatory macrophages when they're given only vehicle that is consistent with age, amplifying the inflammatory macrophages, whereas there's the CD11C positive macrophages fails to increase in old mice treated with CYS3. And consistent with the reduction in the pro-inflammatory macrophages, we see a reduction in IL-1 beta and other cytokine and protection against disease severity. So this reveals the age-specific activity of CYS3. We also performed experiments showing that CYS3 was protected against lethality in old mice. Really, the interesting question is, what is the mechanism of GDF3 SMAD activity? So chromatin accessibility is known to be altered with age, and in particular, large portions of it are more accessible in older organisms. And so this means that there's reduced heterochromatin, so the closed chromatin here, and more euchromatin. Investigators, the UCAR lab in particular, has shown that inflammatory cytokines like IL-1 beta have more accessibility when examining monocytes from old individuals as compared to young individuals. And it's thought that the increased accessibility of the DNA of these genes lead to more transcript. SMAD3 is also a really interesting transcription factor because it acts in a very cell-specific manner, where in each individual cell, it can recruit unique cofactors that allow for specific upregulation of cell-specific genes. And those cofactors can be involved in chromatin remodeling. So we wanted to ask if GDF3 SMAD3 activity is linked to chromatin changes and aging. To address this, she did ATAC sequencing that would allow her to measure the chromatin accessibility in the macrophages. So she took adipose tissue from young control, or old myeloid-specific knockout mice, sorted out the adipose tissue macrophages, and performed ATAC sequencing on them. The overall conclusion from this data was really that GDF3 deficiency reverses age-associated chromatin accessibility in macrophages. And the data that led to this conclusion is, first of all, differential peak analysis, where when she compared young control macrophages to old control macrophages, there was a high proportion of more accessible peaks, so more open chromatin. However, when she compared old control to old myeloid-specific knockout, there was a very small proportion of more accessible peaks, suggesting that the chromatin is closed in the knockout macrophages. When she looked at the peaks from those differentially expressed peaks from those young to old regulated peaks, or the that were down in the old GDF3 knockout, she found about a 50% overlap in the peaks. When she performed transcription factor prediction analysis from those differentially expressed peaks, she found that SMAD4 was a top-regulated transcription factor in the young to old comparison and in the old wild type to old knockout comparison. This was consistent with our hypothesis because SMAD4 is required for SMAD2/3 to actually bind to the DNA to promote transcription. I also just want to point out SUS12, that is a member of the polychrome repressive complex and promotes the trimethylation of H3K27, which normally allows for chromatin closing. More trimethylation leads to a closed chromatin. And we found that GDF3 is regulating SMAD4 specific targets where the transcription start site of IL-1R1 and IL-18R, so the cytokine receptor, are regulated in an age and GDF3 dependent manner. So because the GDF3 knockout macrophages have a more closed chromatin, we wanted to ask if this lead to transcriptional differences in macrophages. We used single cell RNA sequencing to analyze the immune cells from the old wild type and old knockout mice. So this is again UMAP analysis where I've highlighted the different macrophage clusters, and you can see a complete loss in cluster 1 when comparing the wild type to the knockout. We analyzed the transcriptional profile of these different macrophage clusters and found that cluster 1 was a highly inflammatory cluster expressing, interestingly, IL-1R1 and IL-18R1. So those cytokine receptors that we identified in the ATAC sequencing and that are regulated by SMAD4. So there's obviously a lot of interesting data here, but what I really want to focus on is just this question of what are the GDF3 regulated pathways. And so to answer this question, Ina performed pathway analysis on the merged macrophage clusters. And what she found is that the pathway's chromatin modifying enzymes and the pathway methylation dependent heterochromatin formation were enriched in the GDF3 deficient macrophages. And in particular, among those differentially expressed genes, she found the PRC2 complex that is composed of SUZ12, EED, and EZH2. So these 3 proteins together allow for the trimethylation of H3K27, where this is the PRC2 complex, polycomb repressive complex 2, composed of those 3 proteins, causing the trimethylation at H3K27 and leading to the repression or the closing of the chromatin. So this would be what we predict to see in our old GDF3 knockout, where the more open chromatin that is deficient in H3K27 would be more active. So because we had identified a more open chromatin and a transcriptional profile that was consistent in chromatin remodeling, we wanted to ask if there was actually differences in H3K27 trimethylation in the macrophages. And so Ina performed flow cytometry to quantify the levels of H3K27 on those macrophages from young or old mice. So this is the flow cytometry plot to show you the staining of H3K27 as compared to a control fluorescence -1. And when she quantified the mean fluorescence intensity, the old macrophages had reduced H3K27 that is consistent with a more open chromatin. And when she compared the old wild type to the old knockout, the old knockouts had a more H3K27 trimethylation that is consistent with the more closed chromatin in the old knockout macrophages. So this is the majority of the data that supports this model where we think macrophages are making their own signal to cement and cause epigenetic changes, forcing and feeding forward into this inflammatory process, where GDF3 is driving this through the SMAD2/3 pathway, causing a more open chromatin and leading to inflammation, ultimately worsening immune resilience and response to endotoxemia. So now that I've told you that macrophages and the immune system is dysfunctional during age, I want to approach this question of what I think this audience will really appreciate is sort of what is the consequence of the aged immune system on metabolism. And going back to this idea of why we're studying immune cells in the adipose tissue. So this audience is probably very familiar with the idea that the visceral white adipose tissue is an organ that becomes dysfunctional with age. The adipocytes expand, they become hypertrophic, where most older individuals have elevated adiposity. So this is a schematic that is showing the adipose tissue composed of adipocytes and anti-inflammatory leukocytes from a young individual that is insulin sensitive and importantly lipolytic capable. I think of lipolysis as both a negative thing and a good thing, where it can be negative if it's chronically elevated and supporting the release of free fatty acid to cause ectopic lipid accumulation in other tissue. However, lipolysis is actually, we need to turn it on when we want to be able to lose weight or activate the immune system, when we need to generate those energetic substrates to act to provide energy. And in the old individual, the adipocytes become hypertrophic. There's an accumulation of pro-inflammatory macrophages and accumulation of pro-inflammatory B cells and T cells, ultimately just supporting the development of insulin resistance and impaired stimulated lipolysis. And this is really the concept that I want to focus on in this part of the talk. So I want to really re-emphasize this idea that the immune cells in the old white adipose tissue are significantly changing with age, going back to the single cell RNA sequencing from the adipose tissue, where you can see in this UMAP large proportions of T cells, B cells, and macrophages. But there are numerous subsets of immune cells where we can see nearly every subset of immune cell. When I look at the particular macrophages and lymphocytes from the young and old mice, I want to highlight that the macrophages are significantly reduced with age, and the B cells and T cells are significantly accumulating. And so we seem to have this shift in environment where the macrophages are losing their ability to be tissue surveilling and protective, and the accumulation of T cells and B cells may support the inflammatory environment. And just to remind you of what lipolysis is, this field is probably very familiar with the idea of canonical activation of lipolysis, where catecholamines like norepinephrine can signal through the beta-3 adrenergic receptor, driving pKa activation and driving activation of the adipocyte lipases, so ATGL, HSL, and MGL, allowing for hydrolysis of this triglyceride into free fatty acid. This is an important pathway because it allows for obese individuals to lose their visceral adiposity. And what's actually become clear over the last decade or so is that lipolysis can probably be turned on by multiple pathways, including what I'm calling the non-canonical pathway that is activated by cytokines or potentially by bacteria through various receptors on the adipocyte, so the TNF receptor or TLR4. And this can probably activate lipolysis through ERK and maybe also requiring pKa. That seems a little bit murky in the field to me right now. But the important thing is the production of these free fatty acids is critical in generating a productive immune response. And so without ATGL, the ATGL deficient mice have reduced immune response and more lethality to LPS. And this is actually can be quite complicated, right? Because you can imagine that inflammation, either the cytokines or the pathogen, are then acting on the immune cell and on the adipocyte. And there's going to be crosstalk from the immune cell to the adipocyte, potentially regulating lipolysis. But the ability of the adipocyte to turn on lipolysis and give those lipolysis products back to the immune cell is required for the productive immune response. All of this supporting the end outcome of tissue function, the level of morbidity that is seen in the organism, and the survival of the organism. And so the thing that I really want to propose is we need to understand why stimulated lipolysis is impaired in old organism, because it might give us new methods to turn on lipolysis when we need to. So we have done experiments looking at the activation of lipolysis in young and old mice that are treated with LPS to mimic sepsis and give the mice endotoxemia. We see the activation of lipolysis, so phosphorylation of HSL at 4 hours after this LPS time point. And we see that the old mice fail to turn on lipolysis at all. What we've shown is that the cecal slurry model is comparable and can turn on lipolysis in a comparable time point, and the old mice fail to turn on lipolysis at this time point. We also find that serum NEFAs are elevated in young, but not the old. And importantly, a lethal dose of LPS actually fails to induce lipolysis. So just increasing the amount of signaling through TLR4 doesn't turn on lipolysis in old mice. But if we give the old mice a simultaneous signal through the beta-3 AR, we can turn it on. This is important because it indicates that the old adipocyte is potentially functional. It just needs the right signal to be able to hydrolyze those triglycerides. I'm not going to go into all of the data. This was published a year and a half ago, but the experiments that we performed, we first asked really what are the cellular mechanisms involved. So we profiled immune cells. We focused on the B cells because as a postdoc, I had identified that B cells were important in impairing catecholamine-stimulated lipolysis. And so we sorted and transcriptionally profiled the B cell subset. But ultimately, what we did is aged out mice that are completely deficient in B cell to examine the role of B cell in inflammation and LPS-induced lipolysis. So in those experiments, we have old mice that are either wild type or mu-MT mice. So these are completely devoid of any B cell. We aged them out to almost 2 years old and gave them LPS. And this is the data that shows phosphorylated HSL was up, in the old B cell knockout mice. Serum NEFAs were up in the old B cell knockout mice, and the mice were protected from disease severity. We identified that B cells were talking to the macrophages, and this was suggested by data that showed a reduction in the NLRP3 inflammasome, including activation of the NLRP3 inflammasome, indicating that the B cell knockout mice had improved macrophage phenotype, suggesting the macrophages could protect the adipose tissue homeostasis a little bit better, resulting in reduced cytokine and less activation in response to endotoxemia. So this created a model in which sepsis can, or through LPS or through cytokine, can turn on lipolysis. And this is necessary and required in young individuals because it allows for the production of energetic substrate. But in the older organism, where there's an accumulation of B cells that are talking to inflammatory macrophages, there are signals that are inhibiting lipolysis and preventing adipocytes from turning that on, ultimately leading to worsened response during endotoxemia. We think this is important because it gives us new insights into why older individuals have exacerbated mortality. It also highlights an importance of adipocyte immune cell interaction. This project is one of 3 recent publications that really examines the impact of B cells on aging and other cell types. So these are an immune cell that are normally thought of as only responsible for producing antibodies, but this identifies important tissue regulating properties that B cells might have. And so when I try to put together multiple projects from my lab to think about a schematic and how immune adipocyte interaction could occur, it creates this complicated picture, and maybe just to walk you through it. We have focused on age-related immune dysfunction, and I told you about a project that looks at GDF3 promoting chromatin accessibility in the inflammatory macrophages. We'll continue to follow this up by looking at the receptors and the requirement for SMAD3. We look at altered crosstalk and the requirement of IL-1 beta and talking to the B cells, or the B cells crosstalk back to the macrophages. And we look at the downstream consequence of these accumulating immune cells on adipocyte lipolysis. So where the overarching consequence is a reduced energetic substrate, leading to reduced immunity in older people. A few bigger picture questions, maybe to help stimulate some discussion, or this is what I think about in my spare time and how I think about what projects my lab could follow up in the future. But really, so are there immune cells that remain functional? I've talked to you a lot about the ones that become dysfunctional, but if there's still some functioning ones, it could be helpful to just remove the dysfunctional ones, leaving appropriate cells in place to protect the person or the organism. Are there therapeutics that can reverse or remove that dysfunctional immune cell? I mean, everybody wants a drug to chase after because that's the easiest response. Want to recognize that sometimes therapeutic and short-term therapies are not necessarily the most functional. Lifestyle changes are likely to be highly effective, including things like exercise or nutritional changes. But then really what we are in the long term interested in is whether this therapy can improve adipocyte metabolic function and how the consequence of this on the white adipocyte. Within what we've focused on is the gonadal adipose tissue, but within potentially multiple adipose depots. So with that, I would like to acknowledge the people in my lab. Inhua Zhang drove the GDF3 story. Anna really drove the B-cell story and the lipolysis project. And Corbin led the CD8 T-cell and checkpoint blockade story. And I have new students coming up and undergraduate to recognize for their contribution, and numerous collaborators who I would not have been productive in getting these projects out. So with that, I am happy to stop and take any questions.
Thank you, Dr. Camell, for that stimulating presentation. I think we have a couple of questions that are coming in through the QA box. I would like to remind everybody to pose their questions through that box. So the first one comes from Dr. Larissa Constantino. She asks if you can please clarify whether immunosenescence is a normal part of aging or whether it's a pathological process. I know it's a complicated question.
Yeah. No, it's complicated. It's great though. I think this is something that we are actively discussing about in the field because um, the process of immune cells becoming immunosenescent may be required to avoid them becoming overactive. So for a good example is the T cell that becomes exhausted. These exhaustion receptors are upregulated to regulate T cell receptor activity. And so if it wasn't present, the T cell would become overactive, leading to more cytokines and more tissue damage. And so it seemed to be this double-edged sword where it is a normal part of aging, but maybe it's actually a big part of it is preventing pathology, or inappropriately leading to pathology. And I think the complexity of this question really depends on just also follows the complexity of the immune system because there are multiple immune cell subsets that all have different jobs.
I think that is a really good answer to what's
It's a great
honest.
question.
That's honestly a really great question because this applies to many things, obviously, in aging. Dr. Shisaka Kuhlman asks whether if inflammatory macrophages are synonymous with M1 macrophages, and is it in that it's understood that M1 macrophages increase with age in humans?
Yes, I would say with if you accept the limitations that the definition of M1 has, then yes, inflammatory macrophage could be synonymous with M1. Since we study the adipose tissue, the macrophage is encountering more stimuli than an M1, and I think of the M1 as an in vitro macrophage that sees interferon gamma and LPS, so two signals only. And in vitro, the macrophage is encountering tons of lipids, multiple proteins, exosomes, a variety of things. And so I like the idea of flavors of macrophage, but we have so far restricted ourselves to the identification of CD11C as this inflammatory macrophage profile. And this is something that we want to explore a little bit better because it's hard to fully understand it and appreciate the complexity of macrophages without more single cell analysis, perhaps.
Okay.
Yeah, and I would say that M1 macrophages, it does seem like the literature is fairly consistent in measuring M1 macrophages among multiple organs, and those increase with age. But of course, like the flavor of what type of macrophage it is in each individual organ is probably slightly variable.
Thank you. Dr. Chris Wiley says, lovely talk. And he says that the GDF3 data is very interesting and convincing. His question is, you mentioned crosstalk between your projects. So I was wondering if GDF3 or SIS3 based interventions have any effects on other age-related changes, such as lipolysis in the fat.
Yeah. Hi, Chris. Thank you. Um, Yes, so these are great questions. We have done only very preliminary analysis to address this question. We know actually that GDF3 does regulate lipolysis. In our hands, it seemed to impair stimulated lipolysis, but there's some data from Alex Banks' lab at Harvard that suggests it is promoting what I think of as basal lipolysis during diet-induced obesity. So it may be more complex than just a simple off or on answer. Um, I think trying to address the exact role of SMAD3, potentially using the CIS3 therapeutic, could help us get at um, some of the downstream consequences of the SMAD3 pathway. I'm also interested in following up this idea of the activin receptor heterogeneity because there are three or multiple type 1s and multiple type 2 activin receptors, and they can heterodimerize in different ways where they're expressed to different extents on different cell types. And so this helps create the complexity of GDF or TGF beta signaling through SMAD. And I think some of this complexity in activin receptor heterodimerization could support different consequences in each cell type or under conditions like diet-induced obesity or aging in a more healthy manner.
Thank you. Dr. Alice Lichtenstein asks if it says you indicated that LPS-induced lipolysis results in the release of free fatty acids that in turn impact the immune response. So do all fatty acids have a similar effect, or is there a specificity for long-chain fatty acids in the omega-3 or omega-6 family?
Yeah, cool question. I don't know. I would love to find some way to address that. There was one publication that did some in vitro work suggesting that oleic acid
Mm,
was required for B cells to become appropriately turned on. But since it's in vitro, it's hard to know how applicable that is to in vivo, and I think this is something that really needs to be addressed a little bit better.
Thank you. Dr. Shannon Wang says, great presentation and thank you. His question is, during aging, adipocyte size increases and similarly to what occurs in obesity, immune cells accumulate. I was wondering if the age-induced and obesity-induced B cell accumulation occurred through the same or different mechanisms. And in fact, I had a very similar question, so this is great.
Yeah, again, I think there must be some similar mechanism. So for example, something like CXCL13, the chemokine that recruit B cells, is up in both obesity and age. However, one thing that I didn't highlight very well is that the B cell accumulation that we see in age is very dramatic in female mice and it's less dramatic in male mice. So I think there's something additional supporting in particular B cell accumulation in old female mice that is unique from that happening in obesity or diet related changes. And what those are, we know we tried overoptimizing mice and that did not um, reduce the B cell accumulation. So it doesn't seem to be that ovary produced hormones is causing it, but it might be multiple signals leading to the B cell accumulation.
Thank you. Dr. Andrew Greenberg has a couple of comments and questions. He said he's unclear about the significance of stimulated lipolysis because lipolysis increased in obesity, for example, even though stimulated lipolysis decreased. So are there any specific age-related components to that that would make the simulated lipolysis more important? And I think you can also read the comments. There's a series of questions about whether serum fatty acids also play a role. So maybe we can tackle those points separately.
Oh, yeah. So I think my argument would be that stimulated lipolysis is important to be turned on at precise moments when those energetic substrates are needed. And in an infection, that can be defined as a time point from when the pathogen is in the organism. And then the critical thing is it needs to be turned off also. And perhaps I didn't highlight that very well because yes, I think chronically elevated lipolysis would have negative consequences, including more inflammation and ectopic lipid accumulation. Um, so the other sort of piece of data that I tend to think about is the idea that insulin regulated sort of basal lipolysis under fed condition does increase lipolysis to some extent under chronic level, but when under fasting conditions or under giving a stimuli to turn on lipolysis, the activation of lipolysis can be, in our hand, tenfold higher, I think, by measuring serum NEFA level. And so I think the amplitude of lipolysis under what I'm calling stimulated lipolysis by providing appropriate beta-3 AR or cytokine signal has a higher amplitude that it's also important to turn it off under an infection or a pathogen encounter. Perhaps that helped flesh out the picture a little bit better. And what the literature seems to support is at least during a short fast. So what I did as a postdoc is just an overnight fast in old mice, and those old mice were unable to turn on lipolysis by phospho-HSL and serum NEFAs. However, we haven't gone longer. So whether fasting for more than 20 hours out to several days would be sufficient to turn on lipolysis. I think the idea of regulating, my hypothesis is that there are inhibitory signals. The precise hypothesis that we're going after is actually that the B cells are secreting something like IL-10 or TGF-beta, which the adipocyte is also expressing a receptor for. Maybe I can. So this signal from the B cell would be IL-10 or TGF-beta. And so it's not necessarily just the inability of the adipocyte to respond through the beta-3 AR or the cytokine receptor, but there's actually something blocking it and preventing PKA activation and preventing this hydrolysis of triglyceride. And so if we can downregulate the signals from the B cells or turn those off, perhaps we would be able to allow older organism to turn on lipolysis when needed. And yes, okay, so the question about the macrophages
Mhm.
is in our hands in the female mice, they are reduced in numbers, and it does seem like maybe there's some variability among cohorts that may be dependent upon the precise diet that they're aged on or the institution that they're aged in. They do become more pro-inflammatory, and this seems to be very consistent among the literature and among aging cohorts from multiple institutions.
And the last point is about whether saturated fatty acids, which are more pro-inflammatory, I guess, would perhaps have a differential
Yeah,
role. Yeah.
yeah, I would agree with that. And I suppose if the adipocytes from the older organisms are predominantly storing saturated fatty acids, that would make present a model in which they are contributing to the inflammation and the inflammatory environment.
Thank you. That was a great answer to the set of questions. So I think we are at time, waiting to see if there are any other questions that come in. Otherwise, with this, we would like to thank you and thank you again for a great seminar. And thank you, everybody.
Yeah, thank you again. I really appreciated the chance to show these projects.
Thank you.